Have you ever wondered what all living organisms are made of? That's where cell theory comes into play. This fundamental concept in biology asserts that all organisms are constructed from basic units known as cells. It's like the biological version of Legos, with each cell serving as a crucial component in the structure of life. The cell theory introduces three basic principles. First, all living organisms are composed of one or more cells. Second, the cell is the basic unit of life. And third, all cells come from pre-existing cells. This theory didn't just materialize out of thin air. It was the result of the collective work of brilliant minds, scientists such as Matthias Schleiden, who proposed the theory for plant cells, and Theodor Schwann, who extended it to all living organisms, laid the groundwork for this theory. Later, Rudolf Virchow added the final piece of the puzzle by stating that all cells come from other cells. So, cells are the building blocks of all life, but how did we come to this conclusion? The journey towards understanding life's basic units started with three scientists. This trio of inquisitive minds took the first steps in the development of the cell theory. Matthias Schleiden, a German botanist, was the first to identify that plant tissues were made up of cells. His observations laid the groundwork for the cell theory, proposing that plant cells were the fundamental building blocks of life. Next came Theodor Schwann, a physiologist. He took Schleiden's theory a step further, applying it not just to plants, but to all living organisms. Schwann's extension of the theory was a game changer, establishing that the cell is the basic unit of life for all organisms, including animals. The final piece of the puzzle was provided by Rudolf Virchow, a pathologist who emphasized that all cells come from other cells. His contribution solidified the cell theory, giving it a firm footing in science. Their work laid the foundations for the modern cell theory. Building on the original theory, modern cell theory has expanded our understanding of life's basic units. The modern cell theory takes the original concepts proposed by Schleiden, Schwann, and Virchow and enhances them with a deeper understanding of the inner workings of life. The theory begins with the simple premise that all living things are made up of cells. From the mightiest elephant to the tiniest microbe, every organism is an assembly of these basic units of life. Secondly, the cell is not just a building block, but also the structural and functional unit of all living things. Each cell is like a bustling city, with different parts performing different functions, all working together to keep the organism alive. The third point brings us back to Virchow's contribution that all cells come from pre-existing cells through division. This is the basis of growth, repair, and reproduction in all organisms. Fourth, cells carry genetic information. This information, stored in the DNA, is passed from cell to cell during division, ensuring the continuity of life. Fifth, all cells share the same basic chemical composition. This universality speaks to the common ancestry of all life on Earth. Lastly, all life processes, including metabolism and biochemistry, occur within cells. These tiny structures are the sites of countless chemical reactions that power life. But how do scientists study these tiny complex structures? To study cells, scientists turn to an important tool, the microscope. This instrument magnifies the world unseen by the naked eye, revealing the intricate structures and details of cells in all their glory. From the protective outer layer called the cell membrane to the power-generating mitochondria, each part of the cell is laid bare under the microscope's gaze. But modern microscopy has evolved far beyond the simple light microscopes of Schleiden and Schwann's era. Today, we have electron microscopes, capable of magnifying objects up to 2 million times their actual size. These allow us to see the tiniest details of a cell, such as the strands of DNA in the nucleus. Technology has also brought us fluorescent microscopy, where cells are stained with fluorescent dyes, making specific structures glow under UV light. This technique helps researchers pinpoint exactly where certain proteins or molecules are within a cell. In recent years, artificial intelligence has also entered the scene, revolutionizing the way we study cells. With machine learning algorithms, we can now analyze massive amounts of data, comparing thousands of cells at a time. AI can recognize patterns and structures in cells that might be missed by the human eye, providing new insights and speeding up research. Through these detailed observations, we continue to deepen our understanding of life's basic units, the cells.